Welcome everybody to a Striatech hosted journal club. My name is Thomas Münch and I am one of the founders of Striatech. I welcome you to another of our journal clubs where scientists present their projects and data, highlighting the use and the application of our products. Striatech is a dynamic biotech company from Tübingen in Germany. We specialize in neuroscience testing tools, in particular for vision research. The Striatech founders are all experienced neuroscientists in the field of vision research. Today, we welcome Catherine Argyriou as our speaker. Her talk is entitled Developing Retinal Gene Therapy for Zellweger Spectrum Disorder, or ZSD. Catherine holds a bachelor degree in genetics from the University of Manitoba. She did her master's in molecular biology and her PhD in human genetics at McGill University in Montreal, where she still currently is as a postdoctoral fellow in the lab of Nancy Braverman in the Department of Human Genetics. Taken together, she has over 13 years experience in basic and translational research. She's also active in promoting women in the life sciences, being the chair of Women in Bio of the Greater Montreal area. And she is co-inventor on a patent which covers the topic of her talk today, gene therapy for treating peroxisomal disorders. We are very happy to have you as our speaker today, Catherine. Before you start, Catherine, let me remind the audience that you will be able to ask questions throughout the event and also to give a thumbs up to other questions. We will get to those questions at the end of Catherine's presentation. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, but you have to click into the question field before you start typing your question. The Q&A panel on the right-hand side will still be available for a couple of days after this live event. So if you're currently not watching live, but a recording of the event and if Q&A is still available, please make sure to enter your question and your name so that we can get back to you with the answer. We are very much looking forward to your questions. So Catherine, please go ahead with your presentation. Don't forget to unmute your microphone and share your screen. And looking forward to hear your talk.
Okay, great. I hope you can see my screen well. Good morning, afternoon, or evening, everyone, depending on where in the world you are. Thanks to Striatech for inviting me to present today. I'm very happy to be here to tell the story a little bit of a project we've been working on in the lab and with other collaborators for a few years now. And that is the story of developing retinal gene therapy for Zellweger Spectrum Disorder, which is a rare peroxisome disease.
Catherine, you have shared your PowerPoint interface rather than the presentation itself.
Okay. Let me see. Is that better?
No, same as before.
It's the same, you're not seeing the whole presentation?
No, I'm seeing your PowerPoint interface.
Oh, okay, because I put it on presenter view. Let me see what I can do about this. Do you have any tips? Because I'm opening up the...
It's when you choose which window to share, you have to pick the right one.
Yeah. That's my only option. Otherwise, it's my screen, which is not ideal. Because, right, it's the interface, but then if I start the show, does that help?
Now it has to
Good.
show, yes.
Okay. I guess it was the other button that needed to be pressed. All right, so let's... There we go. All right. So for those of you that maybe don't remember your basic biology as well or need a little refresher, peroxisomes are a subcellular organelle. They're a ubiquitous single membrane bound organelle about 1 micron in diameter. Each one of them contains over 50 matrix enzymes that are vital for multiple metabolic pathways. The most well-known of these include beta oxidation of very long-chain fatty acids, alpha oxidation of methyl branch-chain fatty acids, and plasmalogen synthesis, though peroxisomes are also very important for reactive oxygen species metabolism and are mediators, of course, of a lot of other pathways as well. Oh, there's a bit of delay on my click. Sorry about that. The enzymes that do the work in the peroxisome to metabolize their various substrates are called PEX or peroxin proteins. Or sorry, the process is mediated by PEX proteins. So PEX 5 and 7 recognize enzymes destined for the peroxisome in the cytosol, and they deliver them to the peroxisome membrane where they can be translocated into the matrix through PEX 14 and 13. Then PEX 2, 10, and 12 act to ubiquitinate. Pardon me?
Oh, go ahead, I'm sorry.
Okay, act to ubiquitinate PEX 5 so that it can be recycled for additional rounds of import. Now, the removal of PEX 5 from the peroxisome membrane is done by this exportomer complex called PEX 1, 6, and 26. PEX 1 and 6 are AAA ATPases that are bound to the peroxisome membrane through PEX 26. And they use the energy of ATP hydrolysis to remove PEX 5 from the peroxisome membrane. If there is a defect in this structure here, PEX 5 becomes trapped at the peroxisome membrane, and we cannot get additional enzyme import happening. So if there are autosomal recessive mutations in any one of those 13 PEX genes, it results in a Zellweger Spectrum Disorder, which is a peroxisome biogenesis disorder. The incidence of these disorders are about 1 in 50,000. So this affects peroxisome biogenesis, which includes import of peroxisome matrix enzymes, peroxisome division, and new peroxisome formation. It results in multiple enzyme deficiencies that in turn result in the perturbation of several biochemical pathways and multisystemic effects. The clinical severity of the disease corresponds to the effect of the mutation on the PEX protein function. We're focusing here on ophthalmological phenotypes for this presentation. So in the intermediate to milder spectrum of disease, the most common ophthalmological involvements are retinitis pigmentosa, macular atrophy, nystagmus, reduced visual acuity, and reduced electroretinogram or ERG. However, there can be multiple effects throughout the body. Sensorineural hearing loss is very common. There, in more severe situations, there can be white matter defects, cognitive delay, bone defects, enamel defects, liver disease, among other involvements. Uh, 70% of ZSD is caused by mutations in the PEX1 gene. And 30% of these are caused by a point mutation in the PEX1 gene that is a 2528 G to A that occurs at the gene level. This encodes a single amino acid substitution in at the protein level, which is a glycine to aspartate at position 843. So we call this common mutation PEX1 G843D. And patients with this allele exhibit a progressive disease course that could be ameliorated by targeted therapy. And that's just what we aim to do, is use a gene therapy approach to improve quality of life for patients with PEX1 disorders. So what is gene therapy? Gene therapy can mean a lot of things, but in the context of this presentation, it means transplanting a normal gene into cells with a mutated copy. So to do this, we use an AAV, or adeno-associated viral vector, that has encapsulated our therapeutic gene, in our case, PEX1. So this AAV will infect the target cell, so to speak, so that the therapeutic gene will be continuously expressed as an episome in that cell for indefinitely, really. And gene therapy, at least for a monogenic disease, is sort of a good approach for a number of reasons. One is that we get to the root of the disease instead of treating the downstream effects. And second is that it's a single treatment with relatively few side effects and no adherence issues. To test the efficacy, or I guess feasibility, of gene therapy to treat PEX1 G843D, we developed the PEX1 G844D mouse model for ZSD. This model is the murine equivalent of the most common human ZSD allele. Luckily, it recapitulates many of the human disease features, such as growth retardation, abnormal peroxisome metabolites, liver disease, and our focus today, retinal degeneration. Now, we've chosen to target the retinal aspect of this multisystemic disease for a number of reasons. One is that all patients with PEX1 mutations suffer from retinopathy, although they may not have the other tissue involvements. Improving vision would improve overall quality of life, in that it improves communication, learning, mobility, and autonomy for those affected. And the retina is an ideal target for gene therapy in that it's a small compartment, it's easily accessible, and it's immunoprivileged. I will also say that the patient organizations prioritize vision and the eye as a system for therapy. So that definitely fed into the decision as well. For those ophthalmologists out there, I'll tell you a little bit about the retinal phenotype in our PEX1 G844D mouse model. This model has a consistently diminished cone photoreceptor function response measured by electroretinogram. Its rod function diminishes over time, however. It has reduced visual acuity as measured by the optomotor reflex, or OMR. They exhibit progressive photoreceptor loss, so a thinning of the photoreceptor layer over time, but before that, a shortening of the outer segments. Decreased bipolar cell numbers with age, and structural and functional defects in the retinal pigment epithelium. These are new findings that we have discovered that are not yet published, the RPE phenotype. So to sort of overview our proof of concept study testing retinal gene therapy in this mouse model, we designed an AAV8, so that is a serotype 8 adeno-associated viral vector, that will deliver an HA-tagged PEX1 gene driven by a cytomegalovirus, or CMV, promoter and enhancer. We chose to tag the PEX1 protein so that we could easily pick it up in the mouse, because they do have their own endogenous mutant PEX1 protein, so we wanted to be able to distinguish easily. And so we used this AAV8 vector to, well, first we validated the vector in vitro, and I'll go over the results a little bit, and then we delivered it to the mice by subretinal injection. We confirmed successful expression of our vector in vivo. We found that it improved peroxisome biochemical metabolites in the retina, and we also found improved retinal function by ERG. And I'll go into a little bit more detail about that process here. To validate our vector, first we used a PEX1 deficient HepG2 cell line for our very first studies. We chose HepG2 because it's readily transducible with AAV8. We transduced the cells at two different doses, a lower dose and a higher dose of vector, and found when we performed immunoblot against the HA tag that there was indeed expression of our HA tagged PEX1 protein at the right size of PEX1 occurring in these cells. As a control, we use the same vector backbone delivering a GFP, and we were able to detect GFP in those treated cell lines as well. For efficacy, on the PEX1 deficient cells, we did an assay for peroxisome import. So in control cells with normal peroxisome function, this PTS1 or peroxisome targeting signal exhibits a punctate localization that co-localizes with the peroxisome membrane protein ABCD3. This means that enzymes that have this peroxisome targeting signal, their peroxisomal enzymes, are correctly being imported into the peroxisome where they can do their work. In the PEX1 null cells, we see that this PTS1 is cytosolic as it is not being imported properly into the peroxisomes. However, after treatment with our vector, we find that the cells that are transduced do have this punctate patterning that's indicative of recovered peroxisome import. So we are getting functional PEX1 into those cells. The next step was the in vivo study. So for this, we used our two vectors, so our therapeutic AAV8 PEX1 vector and our control GFP vector, both on the same backbone, and delivered them to mice by subretinal injection. This is a very special technique that not a lot of people can do, and it involves using a needle to create a bleb between the neural retina and RPE in the mouse and deliver the vector there in 1 microliter total. We use the highest dose possible, which were 10 to the 10 vector genomes for our very, very first proof of concept. And we were lucky enough that our collaborator from Dr. Jean Bennett's lab, Jenny Song, who was an expert in this technique, visited our lab and performed these injections. Our experimental design included 3 cohorts. We had a validation cohort that we terminated early at 2 months after injection so that we could really validate that we had expression of our vector by IHC or immunoblot. And then we also did sort of some preliminary studies on these mice. And then we had 2 big treatment cohorts. One was a prevention cohort that was treated at an earlier disease stage before the decline, before we see a big decline. So these mice were treated at 5 weeks of age. And we had a recovery cohort that were treated at 9 weeks of age when we've already started to see a decline occurring in the retinal functions of the mice. So both cohorts had baseline ERGs performed before starting. They had their subretinal injection treatment. A subset had preliminary ERGs 20 weeks or 16 weeks after injection. And then all of the mice had endpoint ERGs at the experimental endpoint, which was 6 or 7 months post-injection. And also endpoint visual acuity measures using their optomotor reflex. Finally, all the eyes were harvested to perform LC-MSMS for peroxisome biochemical metabolites. I will also say that each mouse received the therapeutic vector, the PEX1 vector in the left eye and the GFP vector in the right eye. And we also had non-injected non-injected, mutant controls, and all the control groups in the wild-type littermates. So our first in vivo validation was to see if we could pick up the HA tag expressed in the RPE and the neural retina, and we did. In these flat mounts here by immunohistochemistry, we do see the green HA being picked up in the RPE and a lot more in the photoreceptor layer. We also found, if we did an immunoblot against PEX1, a big overexpression of the PEX1 treated mice. So we definitely are seeing an overexpression of our therapeutic protein occurring in the retina and the RPE. To assess retinal function, we use the ERG, or the electroretinogram. And what this technique is that we shine light of different or increasing intensity into the eye of the mouse, and an electrode sitting on the cornea is able to detect the amount of signal produced, I suppose, by the mouse retina, and it's indicative of how much signal is actually being transduced, or the response of the retina to the light signal. And we do two measurements. One is scotopic, which is under low light, and it measures rod or mixed rod cone responses, and photopic measurements, which are high light or measure the cone response. And we were lucky to have a colleague at our facility, Dr. Pierre Lachapelle, and a postdoc in his lab, Anna Pelosa, that taught us how to do the ERGs and how to quantify them. So in our first validation cohort, 2 months post-injection, we were able to see a nice expression of our HA tag along the retina that corresponds to the normal endogenous sites of PEX1 expression in the mouse. GFP expression was more diffuse as expected, and we did see various levels of expression sort of across the mouse retina as well, and very high expression in some zones that were nearest the injection site. The preliminary ERG results, at this point, we started, I will say one thing to note is that for all of the plots coming up, the left eyes are always in blue. These are the PEX1 treated eyes, and the GFP treated eyes are always in green. So and the non-injected eyes tend to be in black. So we're always comparing sort of the blue, which is our treated group, against the green or the black. And so we're starting to see a trend at 2 months of an improvement in scotopic ERG. Our photopic or cone-mediated ERG is getting significantly better on average in the treated mice. And we're seeing an improvement, almost statistically significant improvement in visual acuity. At endpoint, which is 6 to 7 months post-injection, depending on the group, for the prevention cohort, for visual acuity, although there's a non-significant trend of increased visual acuity in the PEX1 treated mutant eyes, but then the ERGs all across all measures significantly improve on average, about twofold on average. For our recovery cohort, we see the same result, a trend but not a significant difference in visual acuity improvement, but an ERG improvement that's about twofold across the board. Finally, we looked at peroxisome metabolites by LC-MSMS spec. What we're looking at here is a C260 lysophosphatidyl This is a marker for very long chain fatty acids, which require peroxisomes to be broken down. So these metabolites accumulate when there is a peroxisome deficiency. And we see that in the prevention cohort, the average very long chain fatty acids, or C26-0-lyso-PCs, decrease in the treated eyes compared to the GFP or untreated eyes, and some of them overlap with the wild type as well. So with this proof of concept data in hand, my supervisor, Nancy Braverman, and I pitched our results at the Americhem Therapeutics knockout competition and won venture capital investment. And then that started the next phase of our journey, developing this therapy to treat vision in ZSD. So what that entailed was a development and redevelopment of a maturation plan. So we planned with the investors the necessary experiments, budgets, and sort of a business model of how to take this therapy forward. We filed intellectual property and patent filings in different countries. Licensing negotiations started between the universities and the investors, which we knew nothing about before this all started. And we now have completed the preparation of a rigorous preclinical efficacy data package. So unfortunately, I am not allowed to show those very nice results that we have been collecting over the last few years, as that information is confidential and we wouldn't want to compromise any intellectual property. But I can give you a very high level idea of what that development plan entailed. So to start with, we engineered a new vector design that is clinically translatable, so more stable, and a design that could actually go into a human. And using that new vector, we correlated vector dose with protein expression and peroxisome recovery in mouse and human PEX1 deficient cell lines. So we established very clear dose response correlations in vitro. We also prepared for expanding more rigorous preclinical efficacy trials in vivo using our mice. So for this, we expanded our visual function measures using the optomotor reflex and the ERG, and I'll tell you a little bit about that, the OMR in the next slides. But for the ERG, we purchased the Diagnosis Solaris ERG to decrease the variance quite significantly, which helped a lot. We identified additional or new histological endpoints to use for recovery in the mouse in both the neural retina and the RPE, which came in very handy. We optimized the biochemical assessment of the retina and the retinal pigment epithelium of the mice. So we were able to dissect those two components out, get much cleaner dissections, and also optimize the way that we analyze the samples, again, to greatly decrease the variance and make the results a lot cleaner. And importantly, we performed an in vivo dose finding study. In our first study, we used a very high dose. We found this was not the optimal dose. So we found the optimal dose of vector to use for our expanding studies moving forward. And then finally, we performed an expanded long-term preclinical efficacy trial using our optimal vector dose and our expanded outcomes. And these are the two new pieces of equipment we purchased. One is the Diagnosis Solaris ERG that helped decrease the variance between among animals for ERGs. We also purchased the Striatech Optodrum and Scotopic kit so that we could have this optomotor reflex testing in our facility. Previously, we had to perform that testing at the facility of a colleague, which meant we could only do one test on each mouse, and it was not ideal. So unfortunately, I can't show you results of treated mice, but I can explain a little bit about how we use the optomotor reflex to measure functional vision in our model. Courtesy of Striatech, we have this beautiful image here. So in short, the mouse is placed on an elevated platform in its testing arena, which is surrounded by computer monitors. The stripe pattern slowly rotates around the animal. If the mouse is able to perceive the stripes, it will trigger a reflex response so that the mouse will track the stripes in concert with the movement. And we can see this through a camera in top of the cage. Optodrome has a software that detects and analyzes this behavior, although you can also score manually, which is oftentimes what I have done. And the stimulus pattern is adjusted so that you're able to determine the threshold of how well the mouse can see or perceive the stimulus. So to show you just a bit of our phenotyping results, nothing with the treatment, unfortunately, but using the optodrome. Here we're looking at spatial acuity in control or G844D mice, and I compared values in the left eyes versus the right eyes. So left eye in blue and right eye in red. In controls, we have these nice high values here. This is at 100% contrast and a slight decrease in visual acuity in our mutants. As we decrease the contrast, so 100% contrast being really black and white, but then 30% being sort of a white and gray and 10% of white and very light gray, so harder to perceive. Of course, we have less visual acuity because it's a harder or lower visual acuity because it's a more difficult test. But we maintain this difference, this average difference between the control and the mutant groups at those different contrasts. I also did a test of just contrast sensitivity, and again, we find a significant decrease between our controls and our mutants. And we find that the average values between the left eye and the right eye are quite consistent as well. We found the same trends, although of course lower values because it's a harder test, under scotopic conditions. So we did also we do have the scotopic kit for this system, which uses filters to create scotopic conditions in the chamber to only test rod function. So in short, for our subsequent preclinical efficacy studies, we were able to do 4 different tests: spatial acuity and contrast sensitivity at both high light or ambient light photopic conditions and low light scotopic conditions. So we added a lot more tests that way. I can show you our expanded study design and how it changed for this second preclinical efficacy trial compared to our first proof of concept. Again, we had a validation cohort that had the vector injected at 1 month of age. These were used just for immunoblot to confirm PEX1 levels and to confirm overexpression of PEX1 in both the retina and RPE separately, and LC-MSMS spec for preliminary values for peroxisome improvement. Our treatment cohort had a baseline ERG at 1 month, followed by a subretinal injection, and they had ERGs and visual acuity measures by the optodrome performed every 2 months until the experimental endpoint of 6 months. At this point, we performed mass spec on the neural retina and RPE. We looked at PEX1 levels on immunoblot and cryosections. By flat mount of neural retina and RPE, we scored RPE morphology, cone morphology, inflammation, and by cryosection, we looked at photoreceptor layer thickness, cone length, and GFAP, which is a marker of gliosis. So hopefully those results will be published soon and you'll be able to read about them. But what I will say is the results from these studies far, far exceeded our initial proof of concept. So we had a remarkable improvement across all of these values for all the treated mice. So what's next? We're now completing our proof of concept data analysis, but we do have a plan with our investors on how to advance the development of this therapy. So coming up soon, we would like to schedule meetings with regulatory agencies, pre-meetings with regulatory agencies to decide the requirements of what tests would be needed to go into patients. These will likely include biodistribution studies in 2 species, which we've started pricing, safety toxicity studies, dosing studies in a large animal, probably mice and non-human primates will be used. We've also started thinking about our clinical trial design. We're getting ready to launch a prospective natural history study of vision in Zellweger Spectrum Disorder. This will solidify or provide the relevant outcomes and time points that could be used in a clinical trial. And we've also begun thinking about vector manufacturing, though that continues to be a big challenge. We have commenced business development with our investors in that we are actively looking for additional capital or a partner to partner with us as we approach these IND enabling studies. And we've also applied for several commercialization grants that would help us advance this project further. So with that, I would like to thank all my lab members, especially Sami Omri, who is the retinal whiz in the lab and does all the structural analyses in the retina, and Armenia Di Pietro, who does all our mass spec for biochemical metabolites. I'd like to thank our collaborators, Joe Hasty at the University of Southern California, Jean Bennett and her lab at the University of Pennsylvania. They provided or generated our vectors, so we are very grateful. And Dr. Pierre Lachapelle and Anna Pelosa at the MUHC, who are the electrophysiology experts. Of course, I'd also like to thank the funding agencies and you for listening. And I guess I could take questions now if there are any.
Oh, there are several. Thank you so much, Catherine, for that presentation. It's really a pity that you cannot show the data because I'm very curious now.
Yeah.
Yeah. Let's dive into the questions. Let's see.
Should I stop sharing my screen? I don't, because I can't see, or do I leave the screen on?
You can just leave. We'll take care of it.
Okay.
Yeah. So how did you select the vector design, serotype, promoter? I guess you did a reselection for your later study, but initially and later on, what were the reasons to do whatever you did?
So we chose the AAV8 vector because our cell type of choice is the photoreceptor cells, and the AAV8 most efficiently transduces the photoreceptors. So we had different evidence in our phenotyping to suggest that the photoreceptor cells are kind of the first site of degeneration that occurs in our animal model, and actually appears to be the first site of degeneration in the humans as well. So that's how AAV8 was just the most efficient serotype to transduce these cells, but it also transduces the RPE, which is another target. For our proof of concept, we use that with the CMV enhancer and promoter, and that was just to choose a ubiquitous promoter that would drive very strong expression in any cell type. For our next generation vector, we have switched to a more stable translatable promoter that's still ubiquitous, but we've stuck with our AAV8 backbone as we still feel that's the most appropriate for the cells that we're trying to target.
Yeah, you mentioned at one point that in principle with gene therapy, you should be able to see lifelong expression of the transgene. Is that something you observe with with the rather short lifespan of the mice with a couple of months? But do you see a decline over time, or is it a stable expression?
The expression appears quite stable, especially where we looked at this a lot more extensively in our latest studies. So at least until the experimental endpoint, which is 6 months post injection, there's still robust expression occurring in the neural retina and RPE in our mice. In a real life situation, I guess in humans, in a real life situation, there's one approved gene therapy that's called Luxturna, and I think it's real world evidence now, so 5 or so years after approval, that patients are still experiencing a benefit to the treatment. So also promising results there for humans as well.
So these peroxisomes and the mutated gene, they're probably expressed in all cells, I would assume.
Yes.
The fact that you see most effects in the photoreceptor and RPE, is that because they're metabolically most active of all the cells? Is that the assumption?
That's the assumption. We are doing a study to look very deeply into the spatial sort of localization of different lipids in different parts of the retina presently. But our theory and others' theory is that, well, first of all, peroxisomes are concentrated at the sort of the base of the outer segment, at that outer inner segment junction. That's where we see an enrichment of peroxisomes and mitochondria for that matter. But the thought is that although they're ubiquitous, the photoreceptor outer segment and RPE have secondarily, the RPE, through its connection with photoreceptors, have a very high metabolic need. So they're turning over very all the time. At least the outer segments are turning over all the time. And they're also exposed to oxidative insult with all the light coming through. So we think that, that the peroxisome dysfunction hits these the most because of that high metabolic need.
And I would assume that's also the reason why you chose subretinal injection rather than intravitreal?
Subretinal injection, yeah, because our target was the outer retina versus the inner retina, and subretinal will hit the outer retina first. Intravitreal is best if you're targeting the inner retina, but I will also say, like, it does, intravitreal also has a downside in that it seems to be a little more inflammatory as well. So it's not the most ideal for translation, but the biggest factor in choosing subretinal was really the target cell type. Yeah.
Mm-hmm. There was a question why 10 to the 10 virus particles is considered a high dose. Why did you say that? And then you also mentioned that lower dose actually is better. What you found out?
That was, considered a high dose because that was the max concentration we could give. So that's, I guess, what I meant by that. So but I think it is still considered a high dose for mice anyways. So for our first proof of concept, we used basically the highest dose we could get. So the vector in its buffer as it was produced from the viral core. And so we just went kind of as high as we could as a first pass. So that's where we got the high dose. It was the highest we could do. But in our subsequent dose finding studies, we tried many dilutions of that maximum dose and found an optimal dose that's actually considerably lower than the high dose that we used in the proof of concept, although there was an effect at almost all the different doses that we tested, but we found the dose with the most consistent sort of robust effect and continued on with that.
Yeah, there was a technical question that I will simply read to you so that I don't make a mistake. So it asks if you performed the injection transsclerally and if you first created retinal detachment and injected the vector afterwards, or if that was done in one step.
It was done in one step. The first injections were transscleral, and the others for our proof of concept, and that's just the technique of the different injectors. And the other one was not. It was the more direct method, not the transscleral. And I believe actually the detachment was created first, and then the vector was delivered. Sorry.
Mm-hmm. Yeah. Okay. All right. Another question here is, how does the HSPEX1 amount and pattern compare to the endogenous gene pattern?
So we see that the pattern matches so the natural sort of expression pattern that we see with PEX1. So we see in our mice, and there are other studies and sort of electron microscopy studies in some human tests that would show similar patterning in the human, but in the mouse we see an enrichment of PEX1 at the sort of in the outer inner segment layer, so sort of just above the outer nuclear layer in that inner segment. And that's exactly where we see the enrichment of our vector delivered PEX1 as well. In terms of amounts, it is a clear overexpression. So at least at our kind of optimal doses, if you perform an immunoblot, you see a higher, considerably higher PEX1 in the neural retina of the treated mice, but lower in the RPE because it's transduced less efficiently. But it's certainly a lot higher. But we do not see toxic effects, that being said. So there doesn't appear to be toxicity even at the high level of PEX1 overexpression, which is good.
Yeah, that's good. Question was, what are the RPE defects in PEX1 mutant mice, and when do they start? And did gene therapy help with the RPE phenotypes as well?
Yes, absolutely. Gene therapy preserved the RPE phenotype. So without divulging too much, because it's unpublished and it's Sammy's great work, but the RPE shows a degeneration that's subsequent to the photoreceptor degeneration. And we only start to see this starting after 3 months of age in the RPE. So at 2 months of age, the RPE still looks very normal, very nice, at least at a structural level in our model. And then it starts, we start to see sort of the appearance of inflammatory markers, and then a slow sort of degeneration of the cells. So they lose their morphology, they become enlarged. Eventually, at an old age, you get loss of cells completely, but or regions completely. But what's also interesting is that the phenotype always follows a very distinctive pattern in our model. But it does appear after we see the photoreceptor outer segment thinning. Yes, gene therapy helped a lot to preserve this phenotype, both the structure and reduction of inflammatory markers.
All right, and the next question, I don't know what the abbreviations mean, so I just read it to you, and hopefully you understand. So in LC-MS, did you look at the levels of DHA in these PEX1 mutants retinas, and was this recovered?
Yes, we did. So there is a DHA is a plasmalogen lipid that requires peroxisomes for its synthesis, and it's also very important for retinal function. We found, though, that although DHA is mildly decreased in our PEX1 G844D mouse model, that no plasmalogens recovered after, or were improved. That being said, they're not that different from wild type, but there was no improvement after gene therapy there, although there is very robust structural and functional improvement. So that was a surprise. But what we think, and especially seeing some work of different collaborators or colleagues in the peroxisome field, is that DHA also there is a strong sort of systemic effect of other tissues for its synthesis, perhaps. And so recovering peroxisome functions just at the localized level is insufficient to recover the plasmalogen/DHA levels in the retina.
Okay. Do you know if there are larger animal models for PAX1 mutations, and do you plan to treat those, or have you treated those in your studies?
There are no larger animal models for any peroxisome disease, including PAX1 disorders. So this does not appear to be a naturally occurring disease in any of the animals that are any larger animals. For the mice, we created the knock-in, but there would not be a large animal model that could be treated. Large animals would only be used for sort of safety, toxicity dosing in the future, but not efficacy. Yeah.
Mm-hmm. Then the next question, does supplementation of the PAX1 mutant mice with, for example, long-chain fatty acids or other peroxisomal enzyme products improve the retinal phenotype?
So supplementation, I guess, would have to be of plasmalogen lipids like DHA. The very long chain fatty acids, we're trying to reduce them. So we want to get rid of those, not supplement them. We haven't tried in our model, although in other peroxisome models, researchers have seen that supplementation, DHA supplementation, does improve the retinal phenotype, but not for long. So in sort of a short window at the beginning, it's almost a delay in the retinal degeneration occurring, but but it would be an interesting thing to try maybe in our model in combination with another treatment to see if you could get even a better effect.
Are the human patients take any dietary supplements?
Some of them do. Some patients do take DHA. Some of them take other vitamins for their, like vitamin K for other organ functions. But that's the extent of it, I believe. Some will have a restricted diet to not sort of not ingest phytanic foods with phytanic acid in them, so that because they're not able to break them down properly. So there are some diets that patients can be on, supplementations or restrictions, but it's by no means curative, of course, of the disease.
Mm-hmm. Have you observed any side effects, both of the gene treatment itself, but also of the retinal injection, the subretinal injection rather? And how long did it take for the bleb to reattach?
So I think by 2 months, it seems that the bleb, we didn't look earlier than that. We kind of waited 2 months post-injection to make sure that we had that kind of robust expression of the vector occurring, which takes some time to kick in. By 2 months, there didn't appear to be any sort of toxic effects in the retina. The bleb had reattached, but there might be. So at the highest dose, there were actually some signs with the GFP vector, not so much the PEX1 vector, some early signs of toxicity that resolved sort of between the 2 and 6 month period. So perhaps there's some sort of initial stress that occurs, initial metabolic stress, but there's no lasting effect. At least even at 2 months, there's no evidence of any toxicity, and the bleb has healed.
Since all of these mice are individually treated with that injection, do you see a wide variation in efficacy? And do you have a way of controlling for, well, let's call it good injection versus bad injection, or versus that the gene therapy itself works well?
No, great. Yeah, unfortunately, there's no good way to control for that. Yeah, it's very true that there are good injections and less good injections. The only way we try to deal with that is by having large sample sizes, because you can't do every outcome measure on every mouse. So you do the functional outcomes. Of course, you have no idea who had a good injection or a less good injection. But then in terms of the, you can look at structural outcomes in terms of, protein expression in some of the mice. So presumably for those mice, you might be able to control for it, but it's a subset, because the rest of the tissues go for processing. And you can only look at kind of one thing per tissue, unfortunately, so it's hard to compare. But we overcome that by just having large sample sizes. Though we did find with our most recent expanded study, the results were very consistent. So the variation that we saw in our first proof of concept, and I think it was really a technical improvement and dose improvement, was greatly reduced. So we actually had pretty nice, tight, consistent values compared to our first study, which we were happy about that.
In your preliminary data, you showed a rather robust reduction in the ERG and more of a rather moderate reduction in the optomotor reflex. Over time, does that decline significantly as well?
So it's interesting in our model that we do see the photopic ERG is sort of always low from the get-go, but the scotopic ERG does decline with age. So in our mice, so the rod response starts off pretty good, and then it gradually declined. And we added C wave as well for RPE in our latest studies, and that kind of followed that slow decline. Interestingly, however, for the visual acuity or the functional vision that we test with the optodrum, that stays pretty consistent over time compared to the ERG. Even, I mean, it makes sense for the photopic because that's consistent, but even for scotopic. So we think like the ERG is sort of an average output of retinal function, but for functional vision, maybe even those remaining kind of pockets of cells that are viable are enough, sort of similar with some of the humans that have an extinguished ERG but are still able to see and get around. So we find that it's not necessarily the visual acuity or functional vision is a lot more stable in our model than the ERG that declines, and we're actually able to improve the functional vision with treatment in our latest data. And with the ERG, we're able to either stabilize or preserve the scotopic ERG, but actually improve the photopic ERG with time. So they don't necessarily follow each other, which is kind of an interesting, unexpected observation.
Yeah, model. They measure different things, and yeah, they go really they supplement each other as a type of measurement. So another question, is the mutant mouse considered a model for cone rod dystrophy, since there is an initial cone defect? Do you plan to try cone-targeted gene therapy at early stages?
So I guess it would be a cone rod dystrophy model, yes, by nature, but our gene therapy targets both the rods and cones. So I guess it's doing both. It's very clearly recovering cone function that we've seen in our latest data. We have sort of a regeneration of the cone outer segments specifically, not just the rod outer segments. So our gene therapy targets all photoreceptors and all RPE, and no plan to target specifically the cones at the moment. With I guess it would be a promoter that is cone specific, with a cone specific promoter, because like we said, there is peroxisome dysfunction in all cells. So we use the ubiquitous promoters to recover function wherever it hits. Yeah.
In your planned clinical trial, are you planning to go for children or adults?
Both. And this is, of course, very hypothetical and very early stage, but I think the plan would be anyone with that meets the criteria, which would be around, imaging or OCT to show how many viable cells they have. So I think it would be open to children or adults, so long as they have enough cells left and so long as they're able to complete whatever outcome measures are selected.
Mm-hmm. Now, given the limited data on the natural history of PEX-associated retinopathy, do you plan deep phenotyping studies, and for how long?
Yes, actually, we've completed some of them. So that's a lot of Sammy's work. He did a big, deeper phenotyping of retinal structure, but we've also been doing imaging mass specs, so lipidomics that are looking at, sort of metabolic or lipid levels at very distinct places throughout the retina, to sort of see. So these studies start very early, before the onset of the structural defects. So at that, 1 to 2 months of age, and then they'll extend to our endpoint. But we've started deep phenotyping all the way from 1 month going to over 1 year of age, and hopefully be getting that data out soon.
Mm-hmm. I wonder, I don't know, I wonder if this question was meant for the human patients, if there is any phenotyping for those, but I'm not sure how the question was meant.
It might be, but yes, so there is, we're now planning a prospective natural history study. We've just completed a retrospective review that's in the submission process, in which we compiled data from many patients in our lab's retrospective natural history study that collects records from participants. So we've sort of assembled all the ophthalmological findings together, but prospectively, yes, absolutely. We're planning a 5-year trial in which participants are followed yearly and will receive a deeper phenotyping than what is sort of standard of care clinically. So they're going to receive a whole battery of tests to help us determine sort of the progression of disease, but also what could be good outcomes to use, and how stable those measures are over a 5-year period.
We're slowly running out of time, but we have only 2 questions left, so let's put them in.
Let's do them.
So do you observe any structural protection of photoreceptors and bipolar cells in addition to the functional improvements in gene therapy treated mutant mice? Yes,
so that's something we were able to see again in our last batch of experiments, but definitely preservation of photoreceptors. So we actually see a regeneration of the outer segment there, and preservation of the layer, the outer segment layer thickness also. So yes.
And then last question: the subretinal injection, the injury that causes to the retina itself, and the detachment from the RPE, if that would not cause more inflammation as and of itself?
It can. So it can cause more inflammation, but in the short term. So that inflammation will resolve normally, and but clearly, so I know we have a model that might be sensitive to inflammation given the peroxisome defect and given their inability to sort of metabolize free radicals and whatnot more efficiently. But clearly we've seen that actually with treatment anyways, the effects of the treatment negate any negative effects of the injection. That being said, though, we do not see more inflammation in the mice injected with the control GFP vector than with or with vehicle, because we also had a vehicle treated group in our last study. So there is not more inflammation in mice treated with vehicle or AV GFP compared to non-injected mice. So so far, no reason to worry about that.
Okay. That's it, Catherine. We're through the questions.
Wonderful.
Thank you so much. That was really very interesting. Thank you to the audience for your participation and the really good questions. I wish you all a good 2023. That was our first journal club this year. And goodbye.
Thank you. Bye-bye.